Good afternoon, everyone. Today I'm going to present my project, Spray-Dry Inhalable Phage Cocktail Powders for Pseudomonas infections. Phage therapy has renewed attention. In this therapy, phages infect and kill bacteria by themselves. Bacteria that infected by phages are unable to regain their viability. By contrast, some antibiotics permit bacteria evolution towards resistance. This picture shows the brief idea of mechanism. First, phages bind to a receptor on the surface of bacteria cell. Phages deliver its genomic content into bacteria, and then phages replicates in bacteria cell, kill bacteria eventually. Because one phage enters a bacteria and replicates, releasing 10 to 100 phages particles after lysis. We only need low dose of phage to kill a strain of bacteria. In addition, since phages consist mostly of nucleic acids and proteins, they are inherently non-toxic. For my study, I'm going to use a combination of those three phages. For those three phages, they have been reported that they have antibiotic effect on bacteria, Pseudomonas bacteria. Therefore, I would like to do further investigation on phage therapy. They came from two families, Myoviridis and Podoviridis. Both of them have tiles which can let phages attach on the surface of bacteria. The reason I would like to use phage cocktail is that there is resistance to phage cocktail can solve this problem. Also, phage cocktail can kill a broad range of clinical strains of disease-specific pathogen. It saves time, and it is convenient for doctors to use this therapy on uncertain strains. For my study, I'm going to develop inhalable phage cocktail powders by spray drying for Pseudomonas infections, and also I would like to investigate if phage cocktail powders can be delivered through inhalable airway to the lung. For my project, here is the method I used. Plug assay is used to test phage viability before and after spray drying process. The spray drying process is used to produce phage powders. After powders produced, I will use laser diffraction to test the particle size distribution and use SEM to test morphology of phages. Finally, I will use NGI to test aerosol performance for phage powders. Moving to the result part, the figure shows the title of three phages before and after spray drying process and after 1 month storage in liquid formulation at 20 degree 50 humidity. Y-axis stands for phage title and axis stands for three phages. Title of phage stored at 1 month was stable, and all three phages remain reliable during the spray drying process. It shows that phages can be protected during spray drying process. The left picture shows that powders had spherical particles. In right picture, the phage cocktail formulation was partially crystalline, and the diffraction peaks at around 66 degree, 19 degree, 24 degree, and 33 degree, which are lucent. Lucent forms crystalline structure on the particle surface and provides protection from moisture-induced powder degradation. The phage cocktail powder were atmosphere powders. The mass of powder increased gradually due to moisture uptake, followed by a decrease within 20 minutes at 60% humidity, because water was expelled from the crystalline lattice. The powders may incline to crystallize if the temperature gap between the storage and glass transition is less than about 50 degrees. As the phage cocktail powder showed a TG of 65 degrees, the recommended storage temperature is below 20 degrees. The aerosol performance of phage cocktail powder was assessed at 2 flow rate by osmohalos with different resistance to airflow. There was a very little powder residue in the capsule. This was probably due to the presence of lucent as a hydrophobic excipient, as it is known to enhance flow and visibility. The FPF value were 45% and 62%. when the powder was dispersed at 60 and 100 liters per minute by the high and low resistance osmohalos. Taken together, this data shows that the powder was dispersible and suitable for inhalation delivery. Overall, Pseudomonas phage cocktail comprising 3 phages was prepared in a stable and inhalable dry powder formulation by spray drying process, and also the formulation showed minimal titer reduction in the phages over a month of storage. The powder was dissoluble using both resistance osmohalos. I would like to thank you for all my group members from ADDG group and my professor, Dr. Kim Chen. Thank you all. That's my today's presentation.